Is going to give about a 50-minute talk on her thesis work, and she'll have some acknowledgements to thank people who have supported her here. And then we open it up to questioning to everyone in the audience and everyone on Zoom too. You can put your questions in the chat, or you can unmute yourself and wave your hand, and we'll try and make sure we see you and ask. So public questioning, when the public, and you're welcome to ask anything. And I will tell you that the easy questions are actually the hard questions. And I've seen many students stumped by something that mom asked at the thesis defense. So, okay, so that's the public questioning. Then we'll have, we'll excuse the audience, and we'll do private questioning of the thesis committee of Allison. And once that's over, we'll party. And that'll be down in Surfside. It'll probably be, given the size of Allison's committee, I'm guessing close to 3 o'clock, somewhere in there. And so that's the procedure. I will now hand it over to Maria Verne, Allison's real advisor, to talk about Allison.
Hello, can you hear me? My name is Maria Verne, as Peter said. I had the pleasure of being Allison's advisor for the last few years while she was at Scripps. And today, so let me first say a little bit about what it takes to be a PhD or to complete a PhD. It's a long process. We only do it because we love it. It's long hours, lots of stress, little money, good friends. And I just want to say that Allison was able to successfully bypass and complete all those ups and downs. And we are here today to hear what she has found as a student. Allison arrived here in 2016 as a Master's of Advanced Studies as a one-year project. She got a master's, and that same year she was accepted as a PhD student. The same year, she went to her first Antarctic cruise to start collecting data for her thesis. And after a year, she passed the exams, which is the official. Although we accept the student 1 year, we officially re-accept the student after the exams. After that, she published a paper. She passed her qualifying exams, and then got another master's degree. By the way, and then she spent the last few years analyzing the data. So today, that's what she's going to present. And I'm here just to tell you a few minutes what has been to get to know Allison as a student and also as a person. Allison came from the University of Washington in Seattle, where she got a bachelor's in biology. And then she worked there at the university and an institutes related to the university for several years. Allison has, if you Google her, she has lots of articles online explaining her path. But the main thing is that she discovered there her love for oceanography and for phytoplankton. And that's, I think, how she decided to explore more and do more research as a graduate student. The first thing that I noticed about Allison was her color hair. I think it was green the first time I saw her, but it's been in many colors. And I have to say, it's contagious. Everyone around her also gets colored hair or a wig. The other thing is that she is really good at costumes. And somehow, as a graduate student, she was able to express her art. And it's always been fun to see the pictures after the fact. On a more serious vein, Allison is what I would call an ocean lover. She really expresses, shares, writes about, talks about the ocean. And here, there, everywhere. So that is always something that, how can I say, it connects connect to people, maybe because I'm also a lover of the ocean, but it's a really strong connection that Allison shares with everyone she encounters. And I would say that everyone that interacts with her leaves that meeting with a little bit more love for the ocean. The other day I was at the airport and I saw this book. And I, being an introvert, I said, oh, maybe I should check it out. But then and making this presentation, I said, I don't think Allison has ever found a stranger. Yeah, for her, everyone is a friend. And one of the ways that she expressed that friendship is through communication. She is one of the most skillful communicators, science communicators that I've known, and she can reach any audience. People that had never heard about the ocean, people that live in the ocean, scientists, students, guests on tourist ships, anyone is the perfect audience for whatever Allison has to say. Part of that communication has been expressed as a teacher. Allison goes every summer to Puerto Rico and teaches high school students and freshman college students about oceanography and about the commonalities between Antarctic and tropical oceans. So that is one of the ways her skill as communicator is expressed. And interesting enough, she can do that in Spanish. As a Spanish speaker, I can say that a lot of us learn English, but it's not that common the other way around. But Allison has made the effort to learn Spanish I think as a way to reach people where they are at. And this is a great expression of that. One of during her PhD studies, Allison has found what I think is her niche, is a great combination of communication and science, love for Antarctica, and that is a citizen science, and she will be talking more about it during her presentation. But I have to say that it's very rewarding for an advisor to see a student that in all these years of efforts has found what it could be a lifelong effort. And finally, Allison has used that data collected in Antarctica and presented it successfully in many meetings, in papers, in presentations. And today she is going to be presenting her results to you. Her thesis is entitled Phytoplankton Community Succession in Relation to Salinity and Temperature Along the Western Antarctic Peninsula, a 4-year study leveraging participatory science.
Introduction, Maria. Let me just get set up for a sec. Oh, wait, I'm using this. Sorry. Hello, everybody on Zoom. I'm going to share my screen. Share screen. Interview. Okay. All right.
Hey,
everybody
hey,
on Zoom can see
Allison.
that, and then I'll angle
Allison,
that.
we're seeing Allison.
And
Can you
Dad,
hear
can
me?
you hear me? Okay. I have Dad mic'd up here. All right. Hello, everybody. Thank you so much, Maria, for that intro, and Peter. It's amazing to see you all here. I have been envisioning this day since September 2017. My time at Scripps began here in this room 1 year prior, learning about policy, economics, and social issues around marine science. And that's where I first met Maria and Peter as one of my first oceanography professors, and I clearly had to keep going with the PhD program. So I'm very excited to share with you what I've discovered about phytoplankton community succession in relation to salinity and temperature along the Western Antarctic Peninsula, a 4-year study leveraging participatory science. And I'm going to be covering a lot in this next less than an hour. And I want to start with this outline to guide us through. I'm going to be going over background of the study region in Antarctica, and I will also introduce the coastal ecosystem as well as the importance of phytoplankton. I then will go through 4 different sections. The first section, I'll discuss what we know from the literature already, building a citizen science program, Fjord Phyto, in the second section. The third and fourth section, I'll discuss the workflow, processing all those samples from the field to the lab to a computational analysis. And the fourth section, I'll spend a bulk of my time discussing the data results and interpretation that came from all that effort, where I'll share about the phytoplankton community succession, as well as the environmental drivers of that succession. And then finally, I'll wrap up with conclusions as well as acknowledgements. So let's dive right into the background. Yes. Oh no. What?
Click swap
Why would that
displays.
be? Share. Does anybody know how to? That's not as exciting to see my notes. What do they see now?
It happened to me once, but now I don't remember how did I fix it. Do you have 2 screens or only one? There.
Oh, now we're seeing it.
Sorry, this is the hybrid model. Yeah, it was okay. 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 Wait, but now I might have just presenter view. Are they seeing the? Yeah, okay. Okay. Whew. All right. We're just breaking the ice. So, okay, everybody's good. We're good. Okay. Let's jump in. So Antarctica might be very remote from most of our minds. It is incredibly far away from this part of the world, but what happens in Antarctica does not stay in Antarctica. It is Earth's air conditioning system, and it's a hub for ocean circulation. The Southern Ocean absorbs 40% of global carbon dioxide, making it one of the most significant carbon sinks on the planet. It's also home to a unique ecosystem rich in marine life and biodiversity. And it's also an extreme environment, making it relatively pristine globally and a natural lab for studying environmental change. Conservation decisions made in Antarctica are made using the best available science. However, it is one of the most data limited regions in the world. Because it's such a harsh climate, it's difficult to operate in, and it's cloud covered, so hard for satellites to see through those clouds all the time. And so international collaboration here is very powerful. Now, I want to focus in on this particular part of Antarctica that the blue arrow is pointing to. This is the Antarctic Peninsula, and this video gives you a sense of what this coastal region looks like with these fjords, mountains coming up out of the water. It's an 800-mile coastline, or 1,300 kilometers, and it's ice covered with glaciers that reach the ocean's interface. Those glaciers bring in a lot of fresh water from melting. Antarctic Peninsula is the 3rd fastest warming region on the planet. So since the 1950s, average air temperature has increased more than twice the global average rate of warming. In addition, the average summer sea surface temperatures have increased roughly 3 times the global average. Studies show that 87% of glaciers are in retreat, and they're putting that fresh water into the maritime ecosystem. What does the ecosystem look like? For those of you who might not have a chance to go down there, it is rich and vibrant in life. And it all starts with the sunlight and the phytoplankton. Phytoplankton derived from 2 Greek words for plant and drifter, and they're microscopic organisms that live in that sunlit layer of the ocean. These phytoplankton contribute oxygen, and they regulate carbon cycles and are the base of the marine food web. They are consumed by grazers. One of the most key species in Antarctica is the Antarctic krill, and those krill depend highly on the timing of phytoplankton. Krill are then consumed by the big megafauna, all the animals we know and love, like the baleen whales that are actually migrating down from the equator to come feed in Antarctica every summer. They're also being eaten by seals and Arctic terns that fly from the Arctic, as well as our beloved local penguins. Now, there's a huge diversity of phytoplankton in the ocean. They come in many shapes and sizes. And if you were to imagine the average width of a human hair, it's about 80 microns. So when I'm talking about large phytoplankton, I'm talking about phytoplankton that are 20 microns and greater, and small phytoplankton are less than that. Now, if you imagine a system that has large phytoplankton, you could imagine there's a high amount of carbon uptake that feeds the krill, and that krill and sinking particles and fecal pellets and dead phytoplankton sink to the seafloor to feed the animals on the seafloor. In addition, they contribute to this food web. Now, the problem is global warming will have fundamental effects on the phytoplankton environment. There will be a reduced sea ice cover during the winter, retreating earlier each summer. The surface waters will warm due to warming climate. Surface waters will also freshen due to the glacial meltwater input. And the surface waters will become more stratified or layered due to the temperature and salinity differences. These changes will alter phytoplankton's exposure to light, nutrients, and metabolic stress responses. These changes will alter the diversity and the size structure of the phytoplankton community, and the community changes will fundamentally affect all those higher trophic levels. So if you imagine that system with the large phytoplankton, now we shift to a system that might have smaller phytoplankton. You've reduced your carbon uptake, you've reduced the nutrient availability for krill. Maybe you'd switch to a more gelatinous-based food web that could contribute to sinking particles with the fecal pellets, but it might reduce the amount of nutrition available for the other trophic levels. Some of the main observations that are reported in the literature include that the community is shifting away from large phytoplankton, transitioning to small phytoplankton. There's an idea that the community diversity is declining. Previously unobserved bloom-forming species, or those ones that grow rapidly, are starting to dominate the system. Previously underreported groups of phytoplankton are also becoming dominant. And studies show that community structure and these shifts in phytoplankton are driven by different environmental variables. This is highly complex. It can include a number of combinations of sea ice, light, nutrients, glacial meltwater, and the changes in that salinity and temperature. There are divergent results in the literature with exactly how this will happen and the lack of understanding of the phytoplankton community response to those environmental changes. So this brings me to my first section. Those statements that I just shared, how do we know those statements? Where did that come from in the literature? For this section, I'd like to thank my co-authors, Alyssa, Freya, and Martina. We've been working on this chapter, hopefully to publish. So I looked at over 245 publications to try to understand where these statements came from. And I'm just going to summarize what I found. Spatially, I'm showing a map of the peninsula, and in the blue dots are different locations that vessel-based research happens. In red, those are where the national Antarctic programs are from Argentina, the Polish station, the United States station, and the British
Allison, we can't hear you anymore online.
Okay, can you guys all hear me on Zoom now? Get rid of the light.
Yes.
Is everybody good? I thought I did.
Yes, we can hear you.
Good, okay. Oh my gosh. Can I get rid of the banner? Okay. Maybe, but you can't see Zoom controls here, so. Okay. So, the methods. The methods of quantifying the community differ between studies. So a lot use microscopy, others use pigments or satellites, and there's a new, since the 2000s, DNA sequencing has become a new method. So the resolution of the taxonomic level you can get to of phytoplankton varies, and it actually makes it very difficult to compare the results. So why are the results to date so divergent? I think that it's because much of the uncertainty is due to the spatial and temporal undersampling. So if you have highly resolved time series, meaning you can cover that span of time, they're sparse in their spatial coverage. And if they're highly spatially resolved, meaning they can cover a large area, they're sparse in the seasonal coverage. So this led us to ask the question, is there a way we can expand the spatial and temporal sampling coverage along the peninsula? This brings me to section 2, building the citizen science program, Fjord Phyto. And this work has been published in the Oceanography Journal, and I'd like to thank my co-authors for helping me put this together. So there are many maritime stakeholders in Antarctica. There are 33 national programs from around the world, and you can see here on a map of the peninsula in yellow, those are the year-round stations, and in red, those are summer-only stations from the Antarctic facilities. And then in blue, I've marked the one United States station, Palmer. There's difficulty for polar scientists to access Antarctica, to gain station access, but also to the United States has 2 icebreakers at the moment. There is also a thriving community of tourism in Antarctica. So IATO, or the International Association of Antarctica Tour Operators, was established in 1991. And it contains at the moment volunteer members of 100 private sector operators from around the world. They operate in Antarctica from November through March every single year. And this is an untapped potential with ships of opportunity, some of which I've shown at the bottom panel here. And there's also an increased public engagement that can happen with sampling efforts. So when I talk about this sector, I'm talking about the expedition vessels, the expedition guides that are hired to work on those vessels, and then the travelers that come and go. So the expedition cruise sector provides public engagement and increased sampling efforts. This is showing the number of visitors or visitations from 1992 all the way to 2022, 2023. Those that made cruise only voyages, and then those that set foot in Antarctica making landings. When I started in 2016, there were 33 vessels in contrast to the 2 US vessels and 47,000 visitors. Just in the last couple of years, this last season, there were 122,000 individual private sector travelers in contrast to 10,000 individual National Antarctic Program staff, or those that can go down from all the nations. So this is an incredible community to engage with. And this is showing again a map of the peninsula with all the different locations vessel-based activities can occur. So this is a huge opportunity here. In this next year, there will be 62 expedition vessels, and the United States is going down to 1 icebreaker. So how can we best connect with the expedition cruise sector? We can engage through citizen science. Now, you might have heard this word as participatory science, community science, crowdsourced science, volunteer monitoring, or public participation in STEM. Basically, what it means is the practice of engaging the public in a scientific project, and a project that produces reliable data and information usable by scientists that is open to the same system of peer review that applies to conventional science. In 2016, when I came and met Maria, she had access to this world, and together, our powers combined, we conceptualized what this program would look like. It's actually a lot more complex than you think to develop a citizen science program, and we started with the Federal Crowdsourcing and Citizen Science Toolkit. So in this toolkit, they walk you through, I spent a year going through scoping out the problem, designing a project, building the community, managing the data, and sustaining and improving. And a lot of this thinking came through connections with the Association for Advancing Participatory Science. So overall, the program questions, how are melting glaciers influencing phytoplankton in the fjords along the West Antarctic Peninsula? In order to design a project, we discovered that online training is incredibly important, and we partnered with the Polar Collective and the Infiniscope. And we also require in-person training, whether that's going to conferences or on the ships or going to port. I've spent over 300 days in Antarctica, and it wasn't just for fun. It was for also getting an idea of how our different partnerships work and the operations on board and the teams, anywhere from a 12-person sailing out to a 500-person expedition vessel. How can we scale this program that is not using a sampling strategy reliant on electricity or liquid nitrogen or freezing or even Wi-Fi? When we build our community, we connected with the private sector operators, the expedition guides, the travelers who volunteer to participate, as well as polar scientists and other students now. To manage our data and make sure it's rigorous data collection, we have trained, confident, and competent facilitators. Annually, we improve and test our protocols, and we provide user-friendly equipment. The validity and credibility comes from funding agencies, federal funding agencies, being able to publish and peer review articles, presenting at conferences, and putting our data in repositories to the public. We're making it findable, accessible, interoperable, and reusable, and also having this open access transparency. Finally, to sustain and improve, this also includes building awareness around this program. So that involves talking to news media sources and maintaining an active connection on social media with people who've contributed. Now, to summarize the success of this program, we scoped our problem of how are melting glaciers influencing the phytoplankton overall. We've trained over 80 expedition guides. We've provided gear and friendly equipment to 24 vessels. We've partnered over the years with 16 operators, including over 8,000 participants so far. We test protocols and have gathered over 800 unique sampling events and presented at 29 conferences, 8 publications, and obtained funding from the National Science Foundation as well as NASA's Citizen Science Earth Systems program. And finally, publishing this in high-profile news media and obtaining endorsements. So over the duration, we've partnered with these ships, we've distributed the gear for the season, and we've trained the guides. Now, what do we have them actually do? The guides will get a GPS list of coordinates, and they'll get volunteers to go out to a sampling station. On station, they'll collect environmental samples, which could include conductivity, temperature, chlorophyll, turbidity, and light level. They'll collect surface samples from the seawater that are analyzed with microscopy by my colleagues in Argentina. And that's for getting cell abundance, identification, and carbon biomass estimates, as well as oxygen isotope data. And then they tow a phytoplankton net for 10 minutes behind a boat, and that collects molecular data or DNA and RNA, and we can do species identification. And this is all happening in the water that can ground truth and help develop algorithms for remote sensing capabilities. So the sampling efforts for fjord phytospatially, I've shown you this map from the review section. Fjord phytos covered the coastal region in many more of a spatial range at 43 different sites along the coastal region. And temporally, it's now supplemented that coverage from November through March when the ships are down there. And we now have 7 years of continuous sampling. Of course, when COVID hit, nobody went down, so there's a gap in that year. Now, the scientific motivation for Fjord Phyto is that most studies were that snapshot in time, and Fjord Phyto provides repeated sampling at many locations each month for multiple years. The methods of quantifying the community and the taxonomic resolution varied among studies, but this Fjord Phyto provides a species resolution with the largest seasonal DNA molecular data set from the West Antarctic Peninsula. And phytoplankton show high interannual variability, which was poorly quantified. This data allows us to quantify seasonal and interannual variability among species, groups, and the community, which is what I'll talk about now in section 3, the workflow from processing samples from the field to the lab to the computational analysis. Is everybody still okay? Okay. So what are the general questions? In general, I'd like to know which phytoplankton are present during a season and between years. Can we identify phytoplankton communities? Can we identify succession within a season? And is it the same every year, interannually, at community level, at group level, at species level? And how do the species, groups, and community relate to environmental forcings? So again, this is the sampling science boat activity, and I'm only focused on with my thesis, the molecular data, samples that came from the net tow, and the environmental data with measurements that came from the conductivity temperature depth device, or CTD. I looked at 271 DNA samples and 333 CTD profiles from a map along the peninsula showing just the stations numbered 1 through 32. And that's shown in this panel on your right, looking at the coverage of sampling from November through March for those 4 years. And again, I just want to point out there was a pandemic, so sampling got a bit sparse in between the 3rd and 4th year coming back online. So the net tow sample is dragged through the water. That concentrated amount of material is then filtered down on a 0.2 micron filter. That biomass, really concentrating the phytoplankton, is preserved in a tube with RNA later solution. And all those are sent back here to San Diego, where I work in the Allen lab at the James Craig Venter Institute to extract DNA from those samples and then amplify that DNA with polymerase chain reaction. For my thesis, I specifically targeted using a DNA marker or barcode that can tell a gene of interest, the 18S variable region 9, which is one of the universal markers for eukaryotes or phytoplankton. I then submitted the samples for next generation sequencing at the Upper Campus UCSD's Genomics Medicine Center, where the sequencing brings back amplicon sequence variants or just a portion of DNA sequence that can help us identify the taxa that were in that sample. So I used the PROTUS2 database to do the taxonomic assignment. And then you can take the relative abundance of those reads and compare what's in the community and who's in the community through bioinformatics. And then I can do a deeper data analysis and interpretation of what I've been seeing. So in my work of that 18S data set, I pulled out phytoplankton that I considered from various taxonomic levels of class and division. And that includes diatoms, dinoflagellates, the cryptophytes, marine stramenophiles, or what I'll refer to as masts, haptophytes, rhodophytes, and green algae. There'll be a quiz afterwards, so you're all supposed to dress up like phytoplankton. So to go back to those guiding questions, I'm going to work through each one. And this brings me to section 4, the data results and interpretation. This contributes to chapters 3 and 4 of my thesis, and I'd like to thank all my co-authors that have been instrumental in making this work come together in this last year or two. So which phytoplankton were present? The results brought back 65 different species of phytoplankton, and I've color-coded them by their groups so that, for instance, in the diatom group, all of the diatom species are shades of green, dinoflagellates, there's shades of blue, and masts, there's shades of orange, and so on. Now, I want to understand, can we identify phytoplankton communities? And what I've done here is shown a heat map And this heat map, you'll want to read along the bottom x-axis. Those are those same 65 species, and I've compared them to themselves down the y-axis. So we're comparing these species. If there's a one-to-one relationship, of course, they will be this darker red color and correlate 100%. So, excuse me, the heat map shows that warm colors have a strong correlation between those taxa, and cool colors have a weak correlation between those taxa, or they don't co-occur. And this brought me to doing a Spearman rank correlation, which is with a hierarchical clustering wards method that basically brings together the similarities between groups with the most amount of difference between groups. Most similarity within groups with the most difference between groups. And the least divisions that came from that are these three. So if you see these three clusters that emerged, this gray cluster is the same set here, and they are strongly correlated with each other. Additionally, this blue cluster, when compared with itself, is strongly correlated, but weakly correlated or negatively correlated to this first cluster in gray. And finally, this third cluster here, when compared to the other groups, has weak correlations with the other groups. So I wanted to see who is making up the most, which taxa are the most represented in these groups. And when I focus in on one cluster, I see that I'm just listing the top 5 most abundant, relative abundance of the taxa that are in that group. And I see a lot of orange, which is a cryptophyte, a haptophyte, and these masts. So I've called this a cryptophyte dominated community, and that is represented in this first group. I then looked at the next cluster that emerged and looked at the most abundant taxa and saw that there were a lot of this green color, or most of the represented taxa in this cluster were a mix of diatoms. So I call this a mixed diatom group. And finally, the third cluster, when I look at the most abundant taxa in this group, I see that there is one diatom that highly dominates the sample. And you can also see that it's a blue ribbon in this cluster, so it doesn't co-occur with any other species or in general. And so I've called this a bloom-forming diatom. There's some dinoflagellates in blue, a bloom-forming diatom dinoflagellate cluster. So the main takeaways from this are that I identified 3 communities. There's a mixed diatom community, the cryptophyte dominated community, and the bloom-forming diatom community. These 3 communities were also observed in previous studies. When I talk about succession, what I'm referring to is the sequence of different species and groups that come after another over weeks to months of a time scale, and that's within one growth season. So you could start your season off with sea ice diatoms, move to a diatom bloom, have mixed diatoms, then haptophytes, and end your season with these smaller phytoplankton and diatoms. Do I see this with the same clusters that I had found? Can we identify this succession at the community level? So the first thing I want to draw your attention to is the cryptophyte dominated community, this gray one. I'm looking at days since the start of the season. So in this, when I talk about the start of the season, I'm referring to November 1st. And I've just linearized time for the number of days after that till March 31st. And I'm looking at the proportion of the reeds that were represented in that cluster. So we can see that the cryptophyte dominated community always begins at a low proportion of reeds in each of these years, but it increases over the season. When we look at the mixed diatom community in blue and the bloom forming diatom community in this red-orange color, we see that in the first 2 years, the mixed diatom community is at the highest relative proportion of reeds. And we see that the bloom forming diatom community is lower than that. In the second year, the mixed diatom community drops off rapidly while the bloom forming diatoms increase. But then in these next 2 years, this relationship breaks down. In these 2 seasons, we have the bloom forming diatom community at the highest reed abundance, and the mixed diatom community is low, but increases towards the end of the season, as well as in this last year. Now, this is interesting because the timing of your sampling would reveal different conclusions based on when you go. So if you went the first 50 days on the summer solstice, December 21st, you might sample a high proportion of mixed diatoms, a lower proportion of your bloom forming, and then low proportion of cryptophyte dominated community. 1 month later, you might sample a bloom forming diatom community and low mixed diatoms with high cryptophytes. Now, if you look at the next year, similarly, the same day, you would get another completely different observation where the bloom forming is high and the mixed diatom is in similar proportion to the cryptophyte dominated community. 1 month later, you might see a similar trend. So the takeaway points from this are that cryptophyte dominated communities increased in the late season. The mixed diatom community and bloom forming community alternated in early and late season, and there's high interannual variability in these patterns. This agrees with previous studies, which found these recurrent communities that occur repeatedly and also strong interannual variability. So this challenges the repeatability of that annual succession concept model where it's just a linear one after the other. Now, this brings me to the question, can we identify succession within a season, and does it fluctuate at the group level, interannually at the group level? Here I'm showing individual samples that were collected and the proportion of the phytoplankton reads per sample, where 100% of the sample has the number of species, again, shaded based on the group color. So all diatoms are shades of green. In blue, this dinoflagellate starts to increase in proportion. So the proportion of the colors is the relative abundance of each of those type of phytoplankton in that year. And I've broken the years up again into early, mid, and late. And that just refers to 50-day increments over time so that I could chop up how I'm looking at these. And you see that in the early season, we have a high proportion of diatoms. And in the late season, we then get an increase in the cryptophytes, mass, haptophytes, and dinoflagellates, with a reduction in diatoms. The next year, when we look at variation between years, we see slightly different pattern. There are still high diatom proportion, but we see a higher proportion of the other groups, the dinoflagellates, mass haptophytes, and this red cryptophyte at the top. And those persist in the mid-season and then dominate in the late season where diatoms aren't as prevalent. We can look over the next 2 seasons and see similarly that each year has a slightly different trend in that the timing of when the different groups are showing up in these samples. And the main takeaway here is that the community had a seasonal transition from the early dominance of diatoms, or these large phytoplankton, and a late community dominated by flagellated groups, which are these smaller phytoplankton. In subsequent years, or in the years that follow after, this transition is not always evident. And so what that might be from it, of course, diatoms are autotrophic. They photosynthesize and use sunlight, whereas the other groups contain species that are mixotrophs. They don't only require photosynthesis to make energy. So this expands the findings from the literature, which stated that diatoms, haptophytes, and cryptophytes are the 3 main taxonomic groups in the coastal regions. I've now seen that dinoflagellates, haptophytes, masts, and cryptophytes are could be equally contributing. In addition, we found that some years diatoms can be late and some flagellated groups can be early. So it's not always this trend. This supports previous studies showing this similar occurrence. But again, there's high interannual variability, which precluded our ability to detect long-term shifts. So with only 4 years of data, I wasn't seeing a general shift to the flagellated group in the community. This brings me to my question, can we identify succession within a community at the species level? So if we look here again at the time, early is in a blue shade, red is in a mid shade, and green is in a late shade. I'm just showing the first 2 groups, cryptophytes and diatoms, and those species are just ordered 1 through 26 here. And I am looking at the point in time at which they were at their peak abundance. And that's what the dot represents. And then the deviation away from that peak in time is the bar, the whisker. And so you can see that even within diatoms, there are species that are early, mid, and late within the season. So there's even succession within the group. When I look at the other groups, I see similarly that while diatoms, most of the species appeared early, there were species in the other groups that could also appear early, and most of the other groups show up mid to late. Now, zoom out to the other 4 years, and by looking at all of the groups now and all the different species, we can see that in general, while diatoms are early in the season, there are some seasons, for instance, this last one, where it seems most of the diatoms appeared late and most of the species in the other groups also appeared late. So there are these shifts happening at species levels within the groups. So within one group, there's the succession at the species level. Timing of the individual species can change each year. So it's not that this one species is always early or always late. And succession between those groups changes depending on when the dominance of the groups occur. Now, how does all of this relate to the environmental forcings? First, we need to understand the environment. So the environmental forcings, this analysis focused just on temperature and salinity of the sea surface. Temperature and salinity are fundamental drivers of oceanographic and biological processes, and they're also correlated and drive other environmental forcings, including stratification or the mixed layer depth, meltwater, nutrient fluxes, and light levels. So when I look at the data that came from these two devices, the CTDs, I'm looking at the average surface 0 to 2 meters, which is where the phytoplankton net samples came from. And I can see from the whole data set that the beginning of the season, we're looking again days since November, on temperature, the beginning of the season starts off the coldest, peaks in the midsummer, and then declines again towards the end of the season. This trend for temperature holds in these nonlinear way in the first 2 years and third year. And then again, because of the pandemic and a reduced amount of samples collected, the trend is not as strong. The salinity in red here, we start the season with high salinity and it's a linear decline to lower salinity over the season. And this trend is maintained in the interannual comparison. Now, just to reorient us, again, temperature can be driven by the light levels increasing for the summer in Antarctica. And then the salinity can also be driven by this, not only the ocean sea ice freezing, but the fresh water that comes from melting glaciers. Those are two influences into this. So how does the environment condition drive the relative abundance of the community, the groups, and the species? When I look at the community level, I see that if I'm looking at the number of species or the richness of the community over days, shown here in the first panel, salinity shown here in the second panel, and temperature in the third panel, what I saw is that there was an increase in community richness over time that was significant. And I saw that there was no significant relationship between salinity and temperature with the community. So the takeaway points is the community richness increases over time, but this contradicts previous studies which reported a decline in the community richness over time with decreasing salinity and increasing temperature. When I look at the group level, I can show here now the different groups in the color bars, and I see that diatoms decrease over time, and the other groups, marine stromatophiles, dinoflagellates, haptophytes, green algae, they all increase over the season, and this was a significant correlation. When I look at the relationship of the groups to sea surface salinity, I see that when salinity is low, we have a low richness of diatoms, but a higher richness of masts. And I found that there was a significant relationship only with the diatom group, the haptophytes, and the marine stromatophiles in relation to salinity. When I looked at temperature, I saw that in cold temperatures, the diatoms were highest, while the other groups in the marine stromatophiles were lowest. But as temperatures increased, diatoms decreased and the other groups increased. But the only groups that had a significant relationship to temperature were the diatoms, haptophytes, and masts, similarly to what we saw in salinity. So the takeaway from this is that only those three groups I saw have a significant correlation to salinity and temperature, and that there were fewer diatom species over time with the fresher, warmer waters, but there were more haptophytes and mast species over time. And this agrees with other studies that observed similar trends in the diatoms and the dinoflagellate groups. But their focus was more on dinoflagellates and cryptophytes being important dominant groups. Now I want to look at the species level relationships to this. So I can break these 7 groups up again into their species. And I'm looking now just at the days since November 1st. If you see a colored dot, that means there was a significant correlation with the timing. And if you don't see a color, that means there wasn't a significance. So the color then, if you see red, it's showing a strong positive correlation to that variable. So for instance, all of the species in these groups that are showing red have a strong positive correlation with time, meaning as time goes on, they're showing up later. When we look at the diatoms, we notice that the species, while some do show up later as well in this red color, there are also correlations that are strong negative correlations in blue, which indicates those species arrive later. When we look at the relationship with salinity, again, we can see that if species in these lower groups have a correlation, a significant correlation, they are showing a strong negative correlation in blue, which would indicate that as salinity increases, or the fresher the system's becoming, there's a correlation with those species. Whereas in the diatom group, again, we see that if you have species responding, they can respond strong negative or strong positive. So they would have species that could thrive in fresh or salty conditions. And then finally, with temperature, when I look at the strength of correlation between the different species, not as many species of the groups are significantly correlated with temperature, but if so, from these lower groups, they are showing in warmer or increased temperature, a strong correlation. And then in the diatom species, again, if there is a relationship that's significant, it's species in cooler temperatures. So the takeaway points here are that not all species have a significant correlation to time, temperature, or salinity. The species that do have a significant correlation respond similarly within their groups, except for the diatom group, which in Antarctica, the diatoms are the most abundant or talked about. And within the diatoms, the species have different responses between time, salinity, and temperature, meaning some thrive as the temperature increases while others decline. So previous studies also reported these species-specific responses to temperature and salinity, and the species level response is important to consider. So this work shows that the importance of that to species level. Previous studies had reported that diatom species and bloom-forming species occurred in all salinity and temperature ranges through the season. So you can see how this starts to get very complex. So the guiding questions, which phytoplankton are present? I found 65 different species from 7 major groups. Can we identify phytoplankton communities? Yes, I identified 3 communities. And can we identify succession within a season? Does it fluctuate interannually at community level? Yes. Group level? Yes. And species level? Yes. So how do these relate, the relative abundance, to the environmental forcings? I saw that there are responses whether you're looking at the community level, the group level, or the species specific level. So how do I tie all this in and think about this complex relationships in a 2D space? We can start by looking at a temperature and salinity grid. And here I'm going to look at the peak relative reed abundance from each of those taxa in their environmental space. When I want to think about time, how do I represent time in this? I'm overlaying, this orange color is going to show days, again broken up in those 50 increments in contour shades of orange. And then the groups will be represented by a dot. So each species will be represented by its group color, and it will be showing where there was the peak, where that taxa was at its peak, what temperature, salinity, and time condition was it in. Ta-da. So here we see that in the early season, we have in this lighter color, the salinity is high and the temperature is low. You can think about moving through time following these contours. So if you imagine the mid-season is up around here, you can also move up through this way, and then you concentrate your temperature and salinity environmental conditions in this darker orange color, which is later season. You can then look at the different groups. So let's start by looking at the diatoms, the green circles that you see here. The different species of diatoms are represented with each of their respective points, but a majority of them are represented in this early season in cold, salty environments. As you move through, you can see that other groups start to be at their peak abundance. And then when you finally come in to this concentrated later season, that's when we're seeing this green algae, the blue, the dinoflagellates coming in, and the masts. So this is looking at the whole data set, but what does this look like each year? Because I just said there's a high interannual variability. This is showing each year, and you can see just by looking at the orange space that in the first 2 years, you had your range of salinity differed and your range of temperature. And then in this final year, it was a very constricted temperature salinity range. But I also see that all of the species are still there. So whether or not they're in a contracted environmental condition or they can expand out into their peak abundance at different temperature, salinity, and timings throughout the season can change depending on which year they're being sampled in. And I want to point out, looking at the early season. So if you look in each year, what the conditions of the salinity, temperature, environment were in this early lighter orange color, you can see that that also varies, and which taxa or species are there can change. So the takeaway points from this are that all species are present, even as salinity and temperature range contracts or expands each season, but they appear at different times. And the diatoms are, again, mostly early, while other groups peak later, and species within a group can also be found in various conditions. So how does the relative abundance of species, groups, and communities relate to the environmental forcings? Another way to think about this, so there's a concept where you can think of called a mandala. And a mandala was introduced in 1979 by Margalef as a way to have a thinking piece of representing phytoplankton succession through time and whatever conditions you're interested in. So can we generalize these findings in a schematic that offers insights into the predictability of phytoplankton succession? I've drawn a mandala that is this diamond shape where I'm putting on one axis the temperature. And remember, temperature wasn't linear, so it goes low to high to medium. And then salinity is represented on the other axis, which is linear from low to high. If we think of representing time at the bottom of the diamond early, and we move through the diamond in time towards late season, we can start to understand where we might see those different phytoplankton groups. So I've placed these phytoplankton groups based on those figures I had just shown before. And you can enter the main succession sequence in the diatom here at the bottom in the early season. Now, if you were to have an environment that had slightly fresher or warmer temperatures, you might enter the diamond in an alternate succession sequence pattern. So if we take that example of this 2017-2018 year, and we were to see through this space how time changes and the communities transition, we can place that and think about it on this diamond space. We're starting with large centric diatoms and moving through haptophytes, ending with these flagellate mast communities. If we were to take a different path, we could see that starting early and going up around, we might end up at this pathway of the mandala, entering through and coming through a cryptophyte, green algae, dinoflagellate, ending in a small diatom community. Similarly, if we take an example from another year, 2018-19, and we move through salinity space along the temperature axis, we might see that we would come from the alternate succession sequence with more cryptophytes and end in a small community of diatoms. Additionally, if we start in the early and move through this time and environmental space, we would end up in this succession sequence pathway. So basically, this is like you can enter the diamond or your environment based on your starting community. And then once you're in depending on your temperature, salinity, or environment, it's like a choose your own adventure of succession. So the main takeaways here are that this new polar mandala is nonlinear, which contrasts to a lot of the other models, which are showing this linear sequence of taxa that come after the other. And this data agrees with other studies that show different years. Sorry, there's studies showing that different years that start with different environmental conditions have slightly different communities which leads to different succession pathways. So this brings me through, we've made it from the background through the 4 main sections where I shared the coastal ecosystem and the importance of phytoplankton, what we knew from the literature, and how building a citizen science program Fjord Phyto has enhanced our ability to look at more samples. I discussed the workflow, and then I shared the data and my interpretation of succession and how the environment relates. So this brings me to my conclusions. My first conclusion is that sampling in partnership with the expedition vessels increases the spatial coverage and the temporal coverage of the near shore, that coastal region, which is important for answering these questions about succession. I've also developed the largest molecular DNA data set, or shown that this is a largest seasonal molecular DNA data set with seasonal coverage. I've only looked at the first 4 years with 271 samples, but we have those 7 years total collected. And succession, I saw that seasonal succession began generally with diatoms transitioning to the other groups with higher community richness than previously reported, but the interannual variability of the exact sequence of the groups or species is high. In the environment, the strength of the response at the community level, the group level, or the species level had a large variability. And this, with significant trends only I saw for diatoms, haptophytes, and masts. So the variability in the environment drives these shifts in the species response. It's reflected at the group and community level, what those succession patterns are. But this is depends on when sampling occurs, can highly influence the conclusions you draw. So about the phytoplankton community trends. Now I'd like to move on to my acknowledgements. In the words of Snoop Dogg, I'd like to thank me for not quitting. But primarily, I'd like to start by thanking my committee and my advisor, Maria Renee and Peter Franks, Andy Allen, Jack Gilbert, Fiamma Strano on Zoom, and Omar Akbari. Thank you guys for being a part of my committee, and for Maria and Peter for being a part of this journey in an in-depth, detailed level, even to our personalities and who we are as humans. And I'll never forget the first day Maria and I met, over a lunch, and 2 hours in, it was like, yes, let's hatch this idea, and my whole life changed because of you, Maria, so thank you. And then Peter, you were my first professor of oceanography, and I remember you asking, what do you hope to learn? And I was a bit bitter coming back to grad school, and I said, something I didn't learn on the job. And I have learned so much from you, so thank you for everything you've done for me. I've learned more than I thought I could never learn on a job. Andy, I remember meeting you in 2015 at the Molecular Life of Diatoms conference in Seattle, and I've been so thrilled to be working with you in your lab. And thank you for opening your doors to your team and being a part of this journey and on my committee. Jack Gilbert, I remember meeting you at the Eukaryote Hits conference in our rapid 1 minute meet and greet, and just thought that was an amazing effort where we're brainstorming what do we know about this world of protists and phytoplankton. So thank you for serving on my committee and also being an inspiration to me. And Fiamma, if you can hear me, I hope I'm not muted. Thank you for Fiamma was also one of my professors and taught me all about ice-ocean interactions. And I've learned so much from you. And so thank you also for having me be a part of your work as well. I've appreciated everything I've learned from you. I can't see anybody on Zoom. And then Omar, thank you for being a part of this in this final year. It's been great to get to know you, and I know our work aligns in a lot of ways, and so thank you for being on my committee as well. I have a lot. I want to thank my lab members. I've been incredibly fortunate to be part of 3 labs. So I'm part of Maria Renee's lab, as well as Peter Frank's lab and Andy Allen's lab. And we have a ton of fun together. And a lot of these people have also become my friends. And so even through the pandemic, we tried to make it silly with Maria's lab. And then, of course, Andy's lab is great for going out or to Padres games at any moment in time. And Peter Frank's lab is great for amazing food and cuisine around San Diego and for like therapy sessions with each other. I want to thank my cohorts. So I was part of the Masters Marine Biodiversity and Conservation cohort, and they've been so dear to me and near to me from that time in 2016 up until the present day. A lot of us have great memories together. And my PhD cohort, which all of them defended years ago. So thanks for being an inspiration for me. I want to thank the Start Blue cohort. We were part of cohort 3, and Start Blue has been an incredible way to rethink how science can be done in different models. And the team has been amazing. Also, my mentors with Start Blue. I want to thank all the departments at Scripps, which I'm sorry, I did not list everybody's name. So I went general, and everybody in administration who I've been emailing back and forth all the time, who's helped with all the administrative work that goes with science and being a student. The dive and boating program has been life-changing, getting on the water and under the water and becoming this ocean person, which, side tangent, I was averse to the ocean. My mom almost drowned. And so when we were young, we never had a connection with the ocean. So for me, I was like, never gonna work in the ocean until I got to Antarctica my first time. And that's when I was like, I am an Antarctic ocean person. So thank you to the dive program and boating program for giving me those skills to be an ocean person. Again, with the Birch Aquarium and the outreach that they do, also cleaning the kelp tank, just accidentally sucking up sea cucumbers and doing the fun holiday dress ups and cleaning the shell of the turtle. That has been amazing. The communications office, thank you for all the support and also giving swag all the time that we can show off Scripps with when I'm down in Antarctica, and for also all the mentoring you've given me to talk to the media. As well as the Design and Innovation Department for the recent Chancellor's Award, and of course the Polar Center, and just all the opportunities that have come from everybody at Scripps. I actually want to thank previous mentors too, because they never thought I'd ever go to grad school, which I never did either. And I've been working in science now almost for 20 years. So I have to thank all my very early scientific mentors, Carol and Jennifer and Christina, and my advisor who told me about Maria, my supervisor, Monica, at the end there told me about Maria and said, I have to meet this woman. So also for my mentees, there's only 2 pictured there, Christian and Anise, but I've had the privilege of mentorship I've received, I've tried to pass on to my own mentees. Thank you to all my collaborators here. There's so many of you in Puerto Rico, also the NASA Globe community, the Hearts in the Ice program up in the Arctic, our social science collaborators. It's just been wonderful brainstorming ideas with you. I feel like this is like what musicians are when they think of a song and then they find somebody to finish the song or do the performance. It's similar in science. Of course, thank you for the funding. None of this would be possible without funding. So we, National Science Foundation, NASA, seed grants, and the Citizen Science Program, the UCSD Chancellor's Inauguration Student Award, Mullen Fellowships, Hooter Foundation, scholarships to go to Friday Harbor, where Maria taught us about fjord ecosystems, and generous donors. Of course, I want to thank the pioneering women that worked in Antarctica, because women were banned from working in Antarctica only, 60 years ago. And so it's been in the United States. So it's been, I have to thank them, because all of this is just built on the people who came before. Oops. Oh, thank you to the Fjord Fido team. We've been growing, and it's amazing to keep seeing more and more people be part of this Fjord Fido program. And they're so, like, all of them make this happen. I want a special shout out to Martina and Elena over this past year, which has been me in the depths, and they've been anchors for me. I literally have thousands of people to thank from the cruise expedition sector. So just imagine yourself pictured here. And I have to thank the IATO Secretariat, past and present, the operators, the managers that even hired me to come on board. Sometimes the travelers, all the travelers who volunteer to participate while they're on vacation. Polar Collective, past and present, Polar Science Community, NASA, Citizen Science. Thank you to those who invite me to come speak at seminars in the British Antarctic Survey earlier this year, at big events at the convention center. And thank you to the media for trusting me to share a story with big media channels. And a special shout out to Bob Gilmore, who Maria knew in the very beginning and has also completely changed my life. And the IATO Secretariat, Damon and Amanda Linus, who said, yes, I can come to one of the field staff conferences to pitch this idea that didn't even exist yet. It was just an idea. So thank you for opening the door for that. Also, expecting the unexpected, like when I ever think I'd be filming with Jeff Goldblum. And thank you for the team, the boat dive crew that facilitated that day on the Beister, and also Shayna, who has magic witchcraft and made this whole thing happen. That was very fun and unexpected. And so I also want to thank everyone who's down to get funky and dress up. I see some of you in the audience today. And then I also want to thank all the people who play outdoors with me and come to endurance events and will just do crazy things at a moment's notice without any training. And thanks, I cannot list all my friends, but there's 4 decades of lifetime of friends that I have accrued, and every one of them is important to me. And thank you to everyone. Of course, my furry friend who passed away 3 years ago, Misha, she was my adventure buddy and why I got outdoors so often. And last, thank you to my family. So my mom, say hi to them, Cindy, my dad, Jim, my siblings, Megan and David, my aunts and uncles, Cindy and Scott, my second mom and family, and my godchildren who are now adults and don't need me to be their godmother anymore. And all my extended family. I love you all dearly. And everyone who's made an impact in my life who I forgot to highlight here, I'm so sorry. And with that, I'd like to say thank you. We take a video or selfie really quick. Oh, they're coming. Okay, yeah, get in it. Come on, Peter. Say hi,
Make Allison cry.
everybody. Oh, what should I?
You need
Oh.
to know that she didn't play.
Well, I'll just say, speaking of Thanksgiving, you were up and looking at all those crafts and trying to figure out what's the rhyme or reason here.
Miracles can happen. Yeah, my mom was noting that over Thanksgiving, I had printed out all of the figures and I was like a detective trying to understand what is the story, like what are the phytoplankton doing? I hope you got a sense that it's complex, but yeah.
You said a great talk, great inspiration of all of it. I wonder if you were, or like, do you have any insight on, you identified 2 kind of groups of diatoms, one that would prefer the colder one, and the other one that would prefer the warm waters. Did you find them being like more species rich one of the other one?
Yeah, good question. So the question was, of the communities of diatoms, I was seeing the ones in higher temperature, or the different conditions in the different diatoms I was seeing. So I saw the one group that had the fresh diatoms in the fresh, warmer condition tended to always be either 4 to 6 different diatom species with 1 to 2 dinoflagellate species, and they were mostly the pennates and Cattosaurus types of genus of phytoplankton. So they were they consistently showed up in that environmental condition and not in the early years. But why?
Like more from a genus, like were fewer OPUs associated with those species, or?
Oh, like the total reads. Wait, sorry, what?
OTUs, like how much of the diversity species that?
Oh, yeah. How much of the total diversity?
Because you show like you show different diversities. And
Yeah.
See like times of the season. Just wondering, that's something that the early bloomers would have higher diversity.
Oh,
like subspecies.
Yeah, within the group. So like within the group of diatoms, diatom species richness and diversity was higher at the beginning than the end. So it was usually only,
Distribution, but like you're integrating species, like the species integrates multiple OTUs. Like, is that reflective? Oh,
yes,
species diversity.
yes, I believe so, because I'm using the ASV as the numbers, the species. Like the number of species is the ASV number.
But you were only showing genera and species.
Yeah.
There is a
Oh,
question,
you mean like, okay, feature.
a question here, and then Kayla has a question online.
Yes. Oh, thanks. Hello.
Super cool. I was just curious, after analyzing all your data and learning about all of the abilities, are there any other parameters like environmental or anything else that you would consider adding to the citizen science data collection moving forward?
The collection, yeah. So the question was, any other parameters added to the citizen science data collection? So I feel we've really tested the limits of what can be collected and the amount of time they have to do the activity. We also add oxygen isotope information. That's one of the samples that's collected. I think there could be more done with the CTD profiles and the temperature salinity structure of the water column. I just focused on the surface for this thesis. But I definitely think that's something that could be on the data analysis side, not necessarily what they collect. Things like nutrients, other things like that are difficult to collect because you have to ensure those samples stay frozen, and we can't ensure that. Once they're collected, there are freezers on board, but then does that sample stay frozen until it gets to us? That's been logistically challenging, which is driven why we sample the things that we sample. But yes, there's a lot. I think the collection might be at its max, but we could take other information. Yeah, thanks for the question. Yes. Jill, or
Have you ever looked at how krill populations have interacted with the diatom population?
yes.
Influence each other based on like their changes throughout the
Yeah, so there's a strong relationship between like krill. One of krill's life cycles relies on the phytoplankton or sea ice diatoms that grow under the sea ice. Also, when the krill become larger adults, they can be in the open ocean water column, and krills have been seen to selectively feed on large centric lipid rich diatoms. And so I think that, well, I haven't personally analyzed anything, but from the literature, what I've read from other people studying that connection, there is definitely an influence. So when sea ice is low, or when those sea ice diatoms aren't available, the krill have less to feed on. The recruitment of krill, like the juvenile krill that can grow over the next seasons, have less food. And so that trickles up through the food web. Less krill, less healthy krill. Yeah.
More salves.
More salves.
Yeah.
Jill.
Another question really go back.
Oh,
Well, why don't
Okay.
we'll do in-person stuff first.
If the locations of the samples, if you time for that specifically, like through the temperature, or if that's an additional variable that potentially something.
yeah. Wait, could you repeat the question? The location of?
Yeah, like if, say, there's 3 different ecosystems that you.
Oh, yeah.
Like if the latitude increasing, maybe
Yeah.
potentially have an impact, or if that was just, if it was a colder temperature, that probably was
Yeah.
lower latitude, higher latitude.
Yeah, so the question was like, regionally along the peninsula, if you have different kind of climates, so to speak, how does that influence the results? So, a lot of the ships don't make it down past the polar circle. And so a lot of that data was more sparse in its ability to get the repeated sequence, sampling. So every time the ships go down there, we like, please collect samples. Same within the north. There's definitely a concentration of samples that are taken at certain parts, even within the expedition fleet. So that would be something to look at. I had attempted that early on, and that's another
Yeah,
effort.
I'm never on one trip that say right guy, so.
Yeah. So that is definitely something too. I think as more people want to participate in this, that will just increase those samples from the other parts that our main early ship partners weren't going to. So it's also driven by where they're going and expanding the footprint on the peninsula. Yes.
I have a question that I've never thought to ask you.
Oh,
Why do you focus on the peninsula? not anywhere else?
Antarctica all is important and unique, but it's very remote, harsh environment to operate in. I know from stories from Maria being down there, putting in moorings and trying to put in instrumentation, you come back and it's gone because the icebergs have ripped it out. So I think just accessibility to the peninsula is easier. There's most of the national research stations are there, most of the infrastructure. Also, the expedition ships go there because it's close to South America, whereas other parts of Antarctica, it's just a lot more resource logistics intensive. And so that's one of the reasons it's one of the least studied regions on the planet. So.
Let's, Kayla, do you want to unmute yourself and ask your question?
Yeah, can you all hear me?
Here she goes.
Can you hear me?
Oops, can't hear it.
Oh, we can't hear you, Kayla.
I can text my
Oh
question.
my gosh. Why can't we hear her?
The volume's up.
Can you type your question?
Yeah, I'm gonna, yeah.
She's typing it. Okay.
About 10 minutes ago. Any other questions from people in the room? Yes.
Questions the other person told you about the locations. Do you do anything different of your data analysis that you do like today? So do you do anything different?
Would I do anything?
Yeah, taking out like the locations. Because the beginning you speak about 3 different
Yeah.
site characteristics.
Like if I were to analyze the location differently now, so one of the journeys I had gone down was to look at the individual sites that had high frequency of sampling and just look at that, because even a site, for instance, there's 2 sites, Cooverville and Danko Island. You can see Cooverville from Danko, and the communities there were completely different in their succession pattern. And it's like, you're just right next door. Why would that be? So I think there's a lot to tease out from looking at the individual site locations. For instance, Nico Harbor is a crowd favorite location where there's a lot of ice flow coming out from all of these glaciers. And so it'd be interesting to really understand how the succession is changing in a more confined fjord system, which builds off of the work Maria has done with that as a model fjord. So, yeah.
And I'd like to point out that Allison made decisions over the last year about what to actually study, and finally decided it was easiest to cluster all these stations in the middle, but there were long conversations among all of us about whether to look at along peninsula transects or cross shore transects and that sort of thing, but that's the next 3 or 4 theses. It's a surprising amount
This
of
is a
work.
recruiting event. Any students would like to, any funders who would like to fund that work.
Yeah, have we got Kayla's question yet? Kayla, type slowly. Oh, here.
Oh.
Might be able to read it.
So there's the concern over shift to gelatinous zooplankton dominated community with climate change. Do you think the phytoplankton communities can be heralds of that shift? And if so, what would they look like? What would the phytoplankton? Well, okay, so the gelatinous community, so like krill have these selectively feed on certain phytoplankton, whereas gelatinous, for instance, that salp that I showed is like a vacuum cleaner. It just sucks up phytoplankton, and so it can actually get clogged as well in the kind of phytoplankton it consumes. And so if there's a shift to a different system, I think the concern is that the grazers of phytoplankton, that because krill are so heavily consumed by all those other animals, it's like a key species. If that declines, then there actually is a lot more work happening in these last few years looking at the shift between these communities. It's just thought that gelatinous animals aren't consumed by anything, even though there are studies showing penguins have consumed salps. And so I just think that is an area that needs a lot more research to really understand the gelatinous community, especially as we're seeing more and more jellies. Like sometimes you'll just see a whole bloom of jelly in the surface down there, which I don't think was also observed a lot in the past. Does that answer your question, Kayla? I hope so. The next question,
We
what
also-
I've heard, there's krill fishing there. Is there all altering the ecosystem in any way. Yeah. So there's krill fishing in Antarctica. It's managed. So CAMLAR is a body that governs the Antarctic marine living resources and sets a limit on how much krill is able to be taken out of the environment. Antarctica is unique in its policy in that it's a preventative approach. So it's at a lower target than what would be a normal threshold for fisheries around the world. And so they're a precautionary approach. That's what it's called. So, but there is a lot of debate on whether this is really an important thing for humans to be extracting out of the environment. And while Antarctica's decisions are made using the best available science, as I've demonstrated even with phytoplankton, there's a lot of limited science that's going into feeding into these numbers. So it's a very active conversation. 3 questions from Nina. Oh my goodness, nice. So organized. Okay, are there plans to publish the results of the participatory project of the scale in public science communication journals? So plans to publish the results of participatory. So the results that I shared today, I plan to publish in any journal that would be not necessarily a public science communication journal, but like a traditional scientific journal that this data could go into. We also did do an evaluation of the responses of participants, and that is going to be published in a Polar Special issue for the travel community and social science, which discusses the perceptions of people's experience on the human side of this project, or the human interaction side. Is there a potential plan to make the data publicly available as an online searchable library? Yes, we're currently working towards doing that now, cleaning up this data in a format that can be uploaded into public repositories. A lot of this, since the funding comes from taxpayer dollars, it's one of our missions is to make it fair. That's the findable, accessible, interoperable, and reusable, and that's what we're working towards now. So we have some of the data sets already uploaded to the public repositories, and we're going to continue doing that. What is the plan to continue the Third Phyto project beyond this defense? For the benefit of scientists and mankind, and the public. What is the plan? There is a plan, and the plan is to continue. Because it does benefit the science and the public, and just even things we didn't think about. Like, for instance, some of the most heartwarming stories are people who participated. And then I'm pointing out one particular individual, Steve Mandel, hello Steve, changed his entire retirement hobby to be a phytoplankton enthusiast, and then hosted microscope workshops back in Santa Cruz with NOAA and started selling phytoplankton swag and calendars and shower curtains. So like, this project has given so many more inspiring things that we never considered when we just wanted to know how are melting glaciers influencing phytoplankton. Like, this is expanded beyond what we ever thought. So we're going to figure out how to keep that going. Well, I hope.
All right, I think this is a good time to wrap it up, but congratulations. Great job. Okay, everybody except the committee has to leave. We'll meet you down in Surfside in 45 minutes, let's say, somewhere around there.
Unless I get a front row seat
Yeah.
to the proceedings. You need to give me my microphone.
Oh, good choice.
Otherwise, I'd be listening to everything there.
Yes.
I could have just snuck that in, couldn't I?
Yes, you could have. Why don't you be-
You could have been eavesdropping. Oh, thank you.
Oh, you can see what it's- Okay.
Oh, and I told Fiona I would call her in on a different Zoom.
Shall I start with this one?
Yeah.
All right. Why don't you
Oh,
say
yeah.
goodbye to people?
Bye, everybody on Zoom. Thanks for coming.